When myocardial infarction occurs, there are massive amounts of cardiomyocytes die. So to compensate for the loss of the contractile tissue, dead cardiomyocytes are replaced by scar tissue, which is non-elastic, and therefore it reduces the contraction of the heart. As long term with untreated, uh, the progressive heart failure will occur. So to prevent the the possibility of a heart failure, people design heart patches which may contain cells or drugs to deliver some uh, molecular signaling to promote the functional cardiomyocyte migration and suppress the scar formation. So here on the right side of the screen, there is a symbol models of combining cardiac extracellular matrix and human cardiac progenitor cells into a GLMA within one bioink, and the bioink was printed in an infused pattern of 90-degree grids. And it was shown that with this simple model, human cardiac progenitor cells had pretty high viability and enhanced expression of cardiac troponin. And the vessel also formed after 14 days in the in vivo model. Uh, on the bottom of the screen, showing a 3D printed curcumin releasing heart patch, which combining nanofibrillated cellulose, PGS polymer, and PPI as a conductive polymer, and all of these in a combination help to make a extended release platform to prevent the burst release of the drug. So regardless of what kind of technique, cells, or scaffold, any engineered tissue always needs a vascularization network to keep them alive in the long term after implantation. So here, people propose that if they use some kind of endothelial cells, which has the ability to form blood vessel into the models, it may help to build some vascular network. So here, they combine iPSC-derived cardiomyocytes and HUVEC, which is human umbilical vein endothelial cells in a bioink to aim for vascularized heart tissue. And they can combine those two bioink together in many ratio and many design. But it was found that with Janus design, the vascular network has better construction and better integrated into the heart tissue. Right. So the model on the top of the screen describes a typical combination of three major components in tissue engineering triad, involving pig heart ECM as materials, bio bone marrow mesenchymal stem cells as cells, and signaling with engineered hepatocyte growth factor into a bio 3D printed uh, scaffold. And the prime bone marrow stem cells has a longer survival in the cardiac patch, and the model showed cardiac protection with enhanced the number of viable cells in the MI heart. Because our heart is contractile, so whatever scaffold that is intended to attach to the heart wall should be elastic. So elasticity of the scaffold should be an important parameter of the heart patches to match for the demand of our heart. So here on the bottom of the screen demonstrated a bioprinted model in which PGS polymer and PCR polymer was combined as a hybrid bioink to print an elastic heart patch for preserve the infarcted myocardium. And the patch show preserving heart function, reducing infarct size, and increasing the heart wall thickness. So as they next model, they combine 3D printed heart patch with a subcutaneous drug delivery system, and the 3D printed epicardial device was made from an bioink containing PGS and PCL. As mentioned before, it has a pretty good elasticity, and the device called PERMAT. The study showed that the PERMAT implantation in the pig heart was performed in minimally invasive surgery, and this result has the potential for treatment heart disease in human patients. And a previously published paper indicated that the microstructure and pattern of the scaffold or the heart patch influence the properties of heart patches. Two important parameters, they are the interstrand distance and strand alignment, was identified as significant factors in evaluating electrical, mechanical, and structural characteristics of 3D printed constructs. So therefore, while designing 3D printed models, bioink components and pattern designs are important. So deep factors play an important role in tissue regeneration, including conductivity, modulus, toxicity, and drug release. The ex vivo cardiac muscle models are the primary applications of cardiac tissue engineering. And to obtain a good functional engineered cardiac muscle, we need to care about organized cell alignment. And the scaffold, therefore, should be designed to induce cardiac cell orientation. And luckily, 3D bioprinting technique has that kind of flexibility to help to create many kind of orientation patterns. And it was found that cardiomyocyte growing on a microchannel pattern gelatin hydrogel scaffold showed the obvious spontaneous fitting than those growing on plain scaffolds. And people also try to build to integrate a resistance sensor whose material was made from black nanoparticles to monitor the heartbeat behavior of the cardiac muscle through the change in electrical resistance. Engineered heart is the main target or the final target of cardiac tissue engineering to make a whole heart organ with full function comparable to native tissue. However, even though people have tried to bring several models such as the heart, heart valve, neonatal scale collagen heart, or human cardiac ventricle model, but it's still challenging for printing an intact 3D structure with heart geometry. And there are some other aspects need to be fulfilled, such as combination of multiple cell types, cell-cell interactions, cell orientation, structure with different material, and especially is the survival of the cell after implantation into the model and how they perform the contraction that can integrate it with the heart contraction.